Welcome back to another edition of Research Connection. Joining me today is Dr. Kelly Meiklejohn and Teresa Tiedge. Hello.
Hi.
So let's start with a little bit about you two.
I can start. So my name's Kelly Meiklejohn and I joined NC State in January of 2018 and I'm an assistant professor of forensic science. And so our lab looks at new sequencing approaches to both human and non-human forensic evidence.
My name is Teresa Tiedge and I am a PhD student in the Meiklejohn lab. I have a bachelor's and master's degree in forensic science. So my background is primarily in forensic science and in biology. And so I came here in January of 2020.
Fascinating. And you just recently received an NIJ grant, correct?
Yes.
Tell us a little bit about it.
So it is a graduate research fellowship. And this will provide support for 3 years, which is fantastic. And so this project is looking at non-human DNA. So my background is primarily in human DNA. So this project is a little bit different. And so we're focusing on the stability of biological taxa. And so what we defined as stability was the persistence and the consistency of these taxa. And so the taxa that we're looking at are bacteria, fungi, plants, and arthropods. And so this project will be looking at the stability of these taxa over a period of 1 year from 2 different sites, an agricultural site housed at the CVM, and then an urban site. And we're also looking at using my cat's tail. We're focusing on utilizing different mock evidence items. So we're looking at porous and non-porous, commonly submitted evidence types. And so we'll be collecting soil and dust off of these mock evidence types. And then using DNA metabarcoding, we'll characterize those biological taxa that I had mentioned earlier within these geologic evidence and hopefully identify which taxa or which taxon is the most stable.
And I'm curious, what is the urban site you're utilizing?
Dr. Meiklejohn's backyard.
And anything to add, Dr. Meiklejohn?
Yeah, so this is a really interesting project. So a lot of the work that had been done in the forensics field on the use of biological taxa associated with soils had really focused on bacteria. And it can change over time. So if you think about a forensic scenario where you may collect an evidence sample, and then many months later, they may actually go out and collect what we call a known sample. And so if those communities of biological taxa shift, how does that impact how we can compare these samples for forensic applications? And so we've seen previously that bacteria does modify depending on the conditions and the time of year and things like that, and then also the type of evidence. So this is the first study that'll look at multiple different types of biological taxa, but also over a period of time, and then also different types of evidence, because a lot of the work that had been done to date had been done on surface soils, and very rarely does a single surface soil get submitted as evidence. It's very more common that it's soil adhered to a tire, a shovel, a shirt, and things like that. So if this was something that was to be implemented into casework, we need to know how it works more broadly, because they're not going to implement something that only works with one type of evidence, one type of geologic evidence. It needs to be more broad. And so Teresa's study will hit at that. So it's really going to feel a need that has been lacking in the community. Yeah, and I'll just add one more thing. I think it's so important with forensic projects that it has that applied nature. We want this to be something that's used by practitioners. And so if we don't have that input and dialogue from them when we're preparing a study design, it really doesn't even, we have to make sure that we have that, because it won't hit home at the end of the day. So that's why it was so pivotal to have input from practitioners who work in this field, so we can get some idea as to, okay, would this be a type of evidence you would recover, so that we're making sure that we're really processing material that would be relevant for them.
Well, it sounds like great work. Thank you for sharing.
Thank you.
And best of luck.
Oh, thank you so much.
Thank you.